I'd like to welcome everybody to the Myra Samuels lecture. It's something we do every year. It's an opportunity to bring an outstanding researcher to Purdue, and it's also an opportunity to remember Myra. While I was driving in to work today, I was looking at the trees, and it reminded me of a project that we did with Myra. The trees don't have any leaves on now, so you can see the branchiness. And Myra was very interested in branchiness of trees and other kinds of structures, and developing ways to measure branchiness. And we had a collaborator in the biology department, and Myra insisted that we go over to that side of campus and bring our lunch and sit under the trees and look at the branches. And we had many pleasant lunches doing that. I could fast forward to the present time, and we have a researcher in the department of, it's called MCMP, Medicinal Chemistry and Molecular Pharmacology, and he studies branchiness. He has collaborators in North Carolina that work with Native Americans who have plants that they use in their medicine that help with the branchiness of neurons, and he's developing therapies that would work for Parkinson's disease. And I wish Myra was here to work with him on that. It would be fun. Which brings us to working with applications and our visitor today, Professor Haiyan Huang is a professor in the Department of Statistics. She is also the director for the Center of Computational Biology at UC Berkeley. She has an affiliation with the Biostatistics Department at Berkeley, too. I took a look at her CV, which is incredible. I would encourage particularly graduate students to browse through. Her name is sufficiently unusual. You get her immediately if you Google her first and last name, no problem. The CV is really, really interesting, though. So I looked at the kinds of things that she's done. She's had a large number of grad students and postdocs. I looked at the list of publications, and a non-random sample. I actually looked at the first two publications, and one is from Nature, and the other one is from PNAS, the Proceedings of the National Academy of Sciences. These are very, very distinguished journals. Then I looked, I said, well, this can't be a random sample, but if you go down the list of publications, there are other publications in those same journals, which is, it's very impressive. If you look at what she's done for, as a faculty member, our responsibilities include teaching. She has taught undergraduate students, she's taught master's level students, and she's taught PhD level students. Those are very different audiences, those of you who've taught in those areas. So not only has she taught that, but she's also taught a course, Stat 375, the title of which is The Teaching of Probability and Statistics, which is something that we probably all need. So I encourage you to look at her CV. It's a very impressive document. So to get to the business at hand, the topic of the talk is GeneFishing, a method to reconstruct context-specific portraits of biological processes and its application to cholesterol metabolism. Thank you.
Thanks very much.
Oh, yes. Before, yes, you can say thank you, but we have to give you something first.
All right. That's fine. Well, before I present back to Professor Huang, I'd like to thank the committee that identified a wonderful speaker that's very appropriate and fit for this memorial lecture. The committee is chaired by George and the other members, Min Zhang and Arman Sabagi. Thank you so much. come here. We can take a picture together.
Thank you.
A little close. There we go. That's very nice. Okay. All right.
Thank you. Thank you so much. Thanks for the great introduction, and it's really my pleasure to be invited to speak in this Myra Samuels Memorial Lecture. Actually, after I received Min's invitation, I searched Myra immediately and tried to understand her. And it was really happy to find that she was a graduate student at UC Berkeley Statistics Department, and her advisor was Jerry Neyman. And actually, in the statistics department at Berkeley right now, so our department, we have a seminar series named after Neyman. And our seminar room is also named after Neyman. And that was focusing on applied statistics and probability too. So it was really a nice connection when I find this. And also it's very inspiring to see Myra's great contributions to applied statistics, especially in applications of statistics to life science, like including this popular textbook. So it's my really great pleasure to be able to speak in this lecture series. As just mentioned already, so this is my talk title. So it's related to a very specific application problem. But I think a lot of ideas underlying the method I'm talking here today will be useful generally to a lot of data analysis problems. So before I start talking about the project in detail, I also want to give a very brief introduction on my research group. So I consider myself as an applied statistician, and I'm focusing on developing high-dimensional statistical and computational methods for different types of data. In particular, biological data. So that is the data I have been working most, during the past almost 20 years. So there are a few example projects that are going on in my group. So one is related to the modeling of gene functional interaction on networks. And especially we have been considering the data coming from heterogeneous samples. So this is the area actually I have been working on for many years already. So I started with like microregion expression data, and then went to the RNA-seq data, the bulk RNA-seq data, and these days we jumped into the single cell RNA-seq data. So the other two, so this project here, so that is the integration for data related to disease, genes, and drugs. So this is the direction that's relatively new in my group. I started about 3 or 4 years ago, and by collaborating with a researcher at UCSF. And so basically we are trying to integrate the information that can link disease, gene, and the drugs together for the purpose of making interesting new therapeutic candidates predictions. And so the random heterogeneous polymer sequence analysis here, so this is a collaboration with a professor at material science on campus. So basically I want to show that, I'm a applied statistician. I'm really open, to collaborate with people who are having interesting data and who are at the frontiers, of scientific research. So when we are handling all of these kind of projects, so there are a lot of challenges that will, arise up. And basically, one thing, or maybe the most important thing is related to the low signal to noise ratio in the data. So like these days, everyone is talking about big data, but for the biological field, the big data is kind of in a different sense. So like when we talk about the biological data is big, usually we mean that, we are having a lot of measurements on different features. Like when we talk about gene expression data, we have, thousands of genes. And that is in nature making the data big. But also meanwhile, the sample size could be very limited. So like for example, a lot of, times my collaborator may only give me, some gene expression measures with like 3 replicates or 5 replicates, extremely small. But these days, of course, we can try to expand that sample size, go to maybe 100, 200. But still, most of the time, it's not really that big. So for the single cell data, it's changing that kind of phenomenon to some extent. So like there are thousands of single cells. But meanwhile, when the sample size, increases, is also adding more noise, to the data set. So the noise will be coming from kind of 2 aspects. One aspect is that, like a lot of genome-wide features will be measured, but based on particular condition we are looking at. So the really meaningful features or functionally active features, it's just a few of them. So like when we talk about thousands of genes, like for human, like 20,000 genes or 40,000 genes, depending on how you define it. So for biological condition, the interesting genes for us, maybe just like 20, 30, 50. So it's hard to be beyond 1,000 or a few hundred. So in that sense, so the signals, are very small compared to the noise. And also when we are increasing the sample size, we are also increasing the heterogeneity, in the sample. And that is adding another level of noise. So therefore, in data analysis, so really, we are fighting with this low signal to noise ratio. And our method to tackle problems around that is trying to combine modern and traditional statistical ideas. And when we are doing that, so one thing we need to be careful is about the computational feasibility of the method. So if the data is big and complex, and even though the method is working in theory, but we also need to make sure that it's working in practice. It has to be computationally feasible. So it's difficult to ask a biologist to wait a week, for the results to come up. So my today's talk will be more focusing on the first example project here. As I mentioned, that has been something I worked for many years already. And when we talk about inferring gene association networks, so roughly we can consider that the task will be involving 3 stages. So at the first stage is about the data preprocessing. So even though I haven't worked much on this stage, but this stage is very critical. So it's setting up the foundation for the subsequent analysis, to be valid. And at the second stage, we want to assess gene-gene interactions. So statistically, we consider that is the construction of eyes-weight matrix for genes. But what similarity measure to use? So that will determine what types of interactions we want to capture. And classically, people have been using like a Pearson correlation, Spearman-Rand correlation, or Euclidean distance, to assess some relationship. But we know that those kind of measures will have a lot of limitations if we want to capture some subtle, interaction patterns between genes. So for example, like sometimes, the expression relationship between 2 genes is not linear. And sometimes, the relationship is not pairwise. It's kind of a group interaction. And one is happening in group, so any pairwise interaction could be small. And those kind of marginal correlation measures may not be able to capture that. And sometimes the relationship may only present locally. So locally means that when you are working with a collection of like 200 samples, for example, maybe the relationship is only happening like in 40 samples or 50 samples. So the relationship may not be a global phenomenon for the data you have collected. So some of my work, has been trying to tackle situations like I have described here. And at the third stage, like after you have built the weight matrix, the next step is trying to find genes that are densely clustered. And we call that as the community detection. So a lot of clustering methods have been introduced, including like stochastic block models. But so I want to first give you a brief idea on what kind of job I have done here, and then I will introduce the central research project I'm going to present today. So these are the two examples. Like, when we are talking about we are interested in assessing gene relationships, that traditional method will fail. So what do we really mean? So like in this scatter plot here, so x-axis is the gene expression for one gene, and the y-axis is the gene expression for another gene. So the each dot here basically means that the expression of that pair of genes under a particular condition. So this is the simulation data. Actually, we simulated that 2 time series gene expression data for the genes. So, but when we were doing the simulation, so we call that a partially coupled time series in the way that only in some of the time domains, so like the purple domain here, the 2 genes of the 2 time series are dependent or are related. So like here, there is a positive correlation and there is a negative correlation. But in other time domain here, the 2 genes are independent. So this is corresponding to the real situation that when the genes are interacting, so that interaction is related to time. So at certain time, so the interaction is there, but as time goes by, it may disappear and then it may come back again. But so this scatter plot is basically plotting this data, so these 2 time series. So you can see that even though there are some kind of patterns there, but if we ignore the time, we don't really see any pattern here. And that's the reason that a lot of classical dependency methods will fail. Because for those methods, they consider the dependency is kind of occurring globally. So they were not designed for the situations like I have described here. And so just for this type of relationship, so we develop a new measure using counter statistics, and that's we call T1, T2. And they are much more powerful compared to all of these other popular dependency measures in recognizing this kind of relationship. So if you are interested in more underlying statistics and also theories of this measure, so you can go to that paper. And later, we extended the method here to the situation that we may have genes from different organisms. So like this is from worm, so this is fly. And so both are time series data, measuring the expression of the genes at different developmental stages, in embryos. But since they are from different organisms, so the time will be not mapped perfectly. So there's still some interesting correspondence for the developmental stages, but it's not a perfect, time matching, considering the 2 time series. And in this kind of situation, if we want to measure the relationship between the 2 genes, what we can do? So previously, people mainly have been trying to study the relationship between genes across species by using sequence information. So like they define orthologous genes, and if they see at the sequence level, there's a lot of similarity, so they have some kind of belief on the functional relationship. And in this study here, so we extended this co-expansion measure still based on counter statistics. So the counter statistics is defined by counting some common patterns, of the expression profile. And we are able to handle, a situation like this. Okay, so this one I'm just trying to give you some kind of idea, like when we are developing methods, so what is the underlying motivation and what type of patterns of interactions or relationships we have been trying to find out. And my today's talk is more related to the stage 3. So like if given the similarity matrix, so you can see that the previous work I introduced was really about the stage 2, right? But today's talk is more on the stage 3. Given the similarity matrix, how we can find, the community. So that's showing that a group of genes that are densely clustered. So in particular, this gene method here, we consider it as a semi-supervised because we found that when there's a lot of noise, and it's better to take into account some prior information. And it's also a non-parametric classing procedure and incorporating the bagging ideas. Okay, so this is the outline here. So I will give you some background and motivation, so how the project was started. And I will give you a sketch about the method. and talk about some properties of the method and also the future work. So what's the background? So how the project has started? So we have a long-term collaborator, Marissa Medina at Children's Hospital Auckland Research Institute. So her expertise is really in the cholesterol metabolisms. So we are long-term collaborators, we are friends, and we have been talking a lot. And so one day, she told me that, so we have this kind of data available. So some of the data was generated by her group, but some of the data was just public data. So like this is the data from her group. And this is the data at the time wasn't available yet, but she was planning to generate. And this is the public data. She introduced this data to me because she thinks this is a biological replicate to some extent to the data they have generated. And also there is a public, a GTEx data set here, for gene expressions across different tissues. So with this all of data, she said that, can you do something to help me answer to some extent, like this? So you can see that these questions are very broad questions. So basically, she's interested in finding new genes that may have some functional role in this cholesterol metabolism. So her interests are really just centering around this cholesterol metabolism. And she knows that, for whatever suggestions we are going to make, it's really just a computational prediction, and the actual validation has to be done by the wet lab experiments or through other predicted and observed functional, consequences. So that's how we decided, to start this project. And you can see that to some extent at that point, the biological question wasn't really that specific. So when I was given this data, so the first thing we did, just very naturally, after some kind of preprocessing, so after some preprocessing by removing some not that expressed genes, so we worked with these 14,000 genes. And this is from the data set, they had produced. We just did a very typical, spectral analysis. So in some way, this is very similar, to a PCA analysis. So each dot here representing one gene. So we were projecting the genes into this two-dimensional space. So and this two-dimensional space is constructed by the first two eigenvectors. When we see that, this looks hopeless, right? So we don't really see any pattern, anything. This is just, a kind of a tight, class of genes there. So we told her that, no, we cannot really start from this. So she has to give us more information to guide us, to make the study more specific to some extent. And since this data was produced under the purpose of studying the cholesterol metabolism, so she said, okay, then maybe you can go to the gene ontology, a public database, to find out the genes that are known to be related to the cholesterol metabolism system. And then to see what happens. So we got these 80 genes and just repeated exactly the same analysis. And immediately, we were able to find two clusters. And for this cluster here, 21 gene, so if we look at the very simple correlation matrix here, so this block here is corresponding to these 21 genes. So very strong signal, just by this simple, Pearson correlation. So I think that's a Spearman-Run correlation. So we are able to see, these 21 genes already. So remember that this is exactly the same analysis, and these genes, are part of these genes too. It's just here because there are too many random or noise genes, so the signals will be buried, overwhelmed, right? So this motivates us to do some computational experiments. And for these 21 genes, actually, when we gave the names to our collaborator, immediately she sent us this back. So those 21 genes, almost all of those genes are belonging to this well-known, cholesterol synthetic pathway. So this is a really biologically meaningful group. Motivated by this, so we did this computational experiment. So what we did here, so this is the figure I shown already. So like based on these 80, known cholesterol metabolism-related genes, doing the spectral analysis, but after the projection, we see the nice separation of the two clusters. These are the 21 genes. So all of the red dots here are the same 21 genes. So the difference is that we are trying to increase the number of random genes into the analysis. So like here, we included additional 500 random genes into the analysis, repeated the same procedure. We are still able to see the red cluster. And here, the red cluster is still there, but we have to use the second eigenvector to separate from others. And the separation is not clean as what we have been seeing here. And when we are introducing more random genes, basically, so the true cluster will just be overwhelmed by the noise. So since this is the data, we know what other true signals are. So we are able to go further, like to find out which eigenvector will be useful to identify these red clusters. And I remember roughly like this one, we have to go to like the 8th eigenvector, and this might be the 13th, and this might be something. It's even further. So you can see that in this experimental experiment, so the signals are always there. So the difference is really the noise level. So when we are introducing more and more noise, so the principal components of the key eigenvector here will not be effective to capture the true signals anyway. And even though the true signals are still there, as I mentioned, that further eigenvectors will be helpful. But in practice, when we do not know the true signal, we don't know which eigenvector to look at, right? So this experiment motivates us to think about a procedure that can make the search more effective in the way that it will be handling a search space that's having a nicer signal to noise ratio. So actually, this slide is the most important slide of my today's talk. So if you do not want to pay attention to other things, actually this slide is enough for you to capture the key ideas of my today's talk. So you can see that we are starting with all of the genes. You can imagine this can be like 20,000 genes. And the idea here is that we do not want to work with this as the search space. So we want to divide it into smaller ones. So these subgroups here will be disjoint from each other, so mutually inclusive. So which means that each gene will be belonging to only one of these random subgroups. So like if there are 20K genes here, the size of each subgroup can only be like 200 or even 100. So that is to make each subsearch space smaller. And also, in each subspace, so we want to make the study semi-supervised. So we are adding into the base genes. So adding into the base genes to make the search better focused, and also in the way that we are able to increase the signal level. So like, the base genes here can be like 10 genes, 20 genes, and the random side of the genes here will be 200. So at least you can see that the signal to noise ratio is not that bad. And after constructing this small search space, then we can just do a special clustering. So somehow I think the version of the slide doesn't look that well, but it's still workable. So after doing the special clustering, so the results very likely will be something like this. And what's more interesting to us is that some random genes will be clustered together with these base genes. When this happens, we call these random genes as fished genes. So they have been fished out by these base genes. And to make the study more robust, so we are repeating this random splitting and also spectral analysis 1,000 times, for example. And then we will aggregate all of the clustering results together from different rounds to make the discovery robust. And then for each gene, so we can count, how many times it has been fished out. And that is then divided by the total number of times, we have been repeating this process. And that number is defined as the capture frequency rate. And this is the number we are using to determine whether a random gene should be considered as part of the cluster or not, or they are considered as truly fished out genes or not. So basically, you can see that the input here will be the expression data and a group of genes. And the output is a list of genes that will be ranked, according to these capture frequency rates. And for the genes ranked, on the top will be considered more likely to be involved in this biological process. So in this analysis procedure here, very natural questions people may ask is that first, why we do not do a PCA analysis for just the base genes themselves and find out the projection or the dimension reduction for these base genes and apply the same projection or dimension reduction to all of the other candidate genes and try to do the cluster. So why that kind of method will not be considered here? So the reason is that I want to point out that when we are trying to do dimension reduction or some kind of PCA analysis, so we are trying to find the key principal components that will be informative, to capture some interesting variation in the data, right? But if we do that just on the base genes themselves, so what kind of variations we are capturing? Just the variations within the base genes, right? But in this product here, the variations within the base genes are not really the variations of our interest. So what will be the variations of our interest? So the information that can distinguish the base genes from the other random genes. So therefore, if we want to find the meaningful principal components or the meaningful eigenvectors, we need to consider a matrix of data. That's including both the positive signals and also some negative signals, because we need the contrast between the positive and negative signals for the purpose of making the eigenvectors or the projected space meaningful. So that is kind of the one of the central ideas of our procedure here. And another interesting property related to the capture frequency rate is that, so this is showing that using the data from our collaborator and based on this chylosynthetic pathway, so what the distribution of the capture frequency rates. So here you can see that we have basically two peaks. So one peak is that the capture frequency rate is really low. So for the genes here, and if you look at the number here, so this is the number of genes, so majority of the genes, they were not fished out, or they were rarely fished out. And for a few genes here, they have been frequently fished out. And you can see that once a gene is fished out, so usually it can be robustly fished out most of the time. So very few genes belong in here. So with capture frequency rates between 0.1 to 0.9. And if we just look at, take a closer look at this bin here, like the capture frequency rate between 0.9 to 1, so majority of them are really at the 0.99 to 1. So this is showing that, what really 0.99 to 1 means? Means that we did 1,000 rounds of GeneFishing, and for these genes, among those 1,000 rounds, they have been fished out 990 rounds. So it's a very robust, discovery. And also this is making somehow the cutoff selection very easy, right? So if we have, remember we are able to rank the genes, and we need to apply some cutoff to determine what genes, are considered belonging to that functional group. And that is a challenging question in many statistical analysis, right? So we have to derive the p-values, for example, or some kind of estimation of false discovery rates that can be used to select a cutoff. But here you can see, roughly it means that no matter we put a cutoff here, here, or here, or even here, the results are not really that different. So the results will be very small. The differences will be very small between the results. So that's another nice property of the method here. Okay, so that was the introduction about the method. So now I want to talk about some applications of this GeneFishing method, and hope that through these applications we are able to understand the method better. So in this study, for the application of the GeneFishing, we focused on those 21 genes. So they are belonging to the cholesterol synthetic pathway. And these are some biological facts we have received from our collaborator. So for the application purpose of what we did, so as I mentioned, so the original study of the project was based on this data set coming from our collaborators group. And we also want to apply this method to this public data. So that's involving a lot of samples for different tissues for the purpose of seeing the dynamics of the pathway along different tissues. And we also studied this GeoVadis data set, since it's kind of a biological replicate of this CAP-LCL data. So for the purpose of the checking the reproducibility of the results. So this is first thing we checked, and this is kind of naive validation for the biological meaning of the study. So like we check the genes belonging to different capture frequency rates. So now we are using 900 or 1,000. These are just meaning how many times they have been fished out of the 1,000 rounds. And the y-axis here is showing that for the genes belonging to that bin or with that range of capture frequency rates, how many of them are known to be related to cholesterol? So based on the gene ontology annotation. So this is a very rough validation. And you can see that basically when the capture frequency rates are higher, so for the genes, they are more frequently fished. So the proportion of them belonging to or related to cholesterol is also higher. So this is basically showing that using the capture frequency rate to determine the membership of a gene to the pathway is kind of involving a quite small number of false positives, right? So that's a very kind of initial validation here. And this is checking the reproducibility of our method. So like we focus on these two data sets, as I mentioned that they are roughly biological replicates, but this data was produced much earlier than this data set. So so this public data, the quality, I think, is not as good as the first one. So what we did is that we apply our GeneFishing method to both data sets, and we also applied a few other methods. So for these three methods here, corresponding to this blue, black, and the green curve, so they are really some kind of intuitive method. People may want to try first. So like what's really the blue one, method corresponds to. So this means that, so since we have the base genes and the candidate genes, we can just simply try to evaluate the association or some similarity between the candidate gene and the base gene. And for each candidate gene, we can use the mean of its similarity to all of the candidate genes to rank, to all of the base genes to rank the candidate genes, right? So you can see that for each candidate gene, we'll calculate its correlation, for example, with all of the base genes separately, and then we take an average of those correlations. And we can just rank the candidate genes based on that. So that's a very naive method, but that can be some method people want to try first. And these three curves are corresponding how to summarize the correlations of similarity measures with the base genes. We can use mean or median or the maximum. And the x-axis here is that when we are ranking the genes based on those, we can take the top 100 genes or top 80 genes or top 60 genes. And for both data sets, so we can see that among those top ranked genes, how many are overlapping. So this is a way to check the reproducibility of the method across to biologically replicate data sets. And you can see that just by this simple criterion, the GeneFishing looks better. And so this orange curve here corresponds to this WGCNA. If you guys have some experience with gene network studies, you will know that this is a very popular method for estimating gene networks. But the comparison of GeneFishing to this one here is unfair for this method because this method is totally unsupervised. So it's not making use of any prior knowledge. But GeneFishing method is semi-supervised, right? So we have baked genes. So therefore, the performance of the GeneFishing is kind of better like this. But that comparison is not fair. Okay, and so for the GTEx data, so that data, is interesting in the way that it's having the gene expression data across many different tissues. And it's providing a way for us to see changes of a network across tissues. So the first tissue we worked on is the liver tissue. So why liver? Because that is the major organ impacting, our plasma cholesterol level. So that's why, we worked on this first. And these are the distributions of the capture frequency rates. So this is that, among the 1,000 runs, so like only less than 100 runs, the genes have been fished. And you can see that, look at the y-axis, the scale here, majority of genes are belonging to this. And this is kind of when the capture frequency rates lie in the middle, so like between 0.1 to 0.9. So they have been fished out to some extent, but not that often. And so not many genes belonging to this. And when you look at here, we do have around 60 genes. They have been frequently fished out. And just based on this figure, we took ad hoc cutoff at 0.99. But we do see that it is a reasonable cutoff, based on this distribution. We wouldn't lose much. And using that as a cutoff, we have in total selected, 56 genes. And when we pass those 56 genes to our collaborator, immediately she recognized that many of them are related, to cholesterol metabolism already. So a lot of them are well-known, cholesterol genes. So for her, what's more interesting? She is interested in finding some kind of genes, that she has no knowledge before. So therefore, she purposely selected a few genes among these 56. So for those genes, they were not previously implicated in cholesterol metabolism, and they didn't show any evidence of steroid regulation. So basically, for those genes she selected, like before seeing our result, she wouldn't believe they are related to the cholesterol metabolism at all. So she was kind of, wondering why those genes have been fished out. So I remember she did experiments maybe on 3 genes, selected 3, and among those 3, so this one is this gene, so it's called the GLO1, is particularly, conclusive. So she tried this on 2 different cell lines, and these are the conclusions, she made. And these are kind of a lot of control experiments. NTC is a control gene, and so basically, it's very convincing experimental results showing that for this gene, when it's knocked down, so the cellular cholesterol, ester levels will be increased. And she also found that, this gene will significantly impact transcript level of this SQLE gene, which is a well-known gene, in this cholesterol synthesis pathway. So like even before seeing the result, she wouldn't believe this GLO1 gene would be a gene in the cholesterol metabolism, but her experiments confirmed, our finding. So that was exciting finding both for her and also for us. And then we want to see that if we just apply the method systematically to different tissues, what will happen? So this one shows a very simple correlation matrix for the genes in different tissues. And for the red bar here are showing the baked genes. And when the colors are in the warm, color span, so they means that they are strongly correlated. And you can see that for those baked genes in muscle, they are not correlated at all. So this is suggesting that in muscle, somehow the cholesterol synthetic pathway is not really that active. And when you look at, for tissues like this, so there are also a lot of other genes are closely related, to this synthetic pathway. So this type of analysis, so we did, so since our collaborator is really just expert in terms of cholesterol pathway and also liver, she wasn't able to explain, in depth regarding what kind of the biological phenomenon this may be suggesting. But we feel that, this is interesting in the future if we want to see the changes of the pathways or want to see some kind of crosstalks, between different pathways. And we did some, so this is showing that, for different tissues here, so some genes fished out will be specific to that tissue, but sometimes they also share a lot of common tissues. So this is showing some functional relationships, between the tissues. And we also studied, like, for each tissue, for the fished out genes, so what are the possible functional groups they belong to? So basically, these are the key observations that we have made. So like, for those, steroid metabolism pathway genes, they have been frequently fished out. And this is meaningful, right? Because we have been, the base genes are belonging to the cholesterol pathway. And also the immune system genes have been fished out in great numbers too. And we know that the immune system genes are related to the, heart disease and a lot of other things, and the cholesterol can be playing a big role here. So how many minutes do I have? One? Okay, if it's one gene, then maybe I will skip some of the results. So like, we compared our method with some well-known methods. And in the literature, so again, our method is showing some advantage, but it's showing the advantage in the way we think that because our method is kind of more specific and better designed, and the other methods are kind of trying to integrate a lot of other information. So sometimes when the information is more, it doesn't really mean that it's always good. You want the data that's more relevant, right? And if you are integrating a lot of non-relevant gene information to the study, then the results may be even worse. So there are also some kind of properties about this. So all of these things can be found in my paper, so I will skip that. So the last thing I want to mention is that now we try to extend this to single cell data. So the single cell data is challenging in the way that the samples will be very heterogeneous. And if you want to just assess gene relationship based on all of the single cells without doing any filtering, the analysis is meaningless in many, many situations. And this one, these are kind of the base genes for another well-known, pathway. And so this figure here is also the same analysis. So we had tried to, filter the single cells already. So this is the figure based on like only, I think, 300 single cells. So it's supposed to be informative, but still we are not able to separate the base genes, from a lot of other random genes. And by this one here, we are able to separate them. So what we did. So what we did here is very simple. So out of the about 300 cells, each time we just randomly sample 50 cells of that. And we calculate the correlation based on those 50 cells only. And we repeat that many, many times, and then we take the average of the correlations, get that kind of average similarity matrix, and then we just do the spectral analysis. And now the separation is there. So what this is suggesting? So this means that, exactly, for the single cell data, the relationship between genes is happening only locally. It's only happening in a subset of the single cells. But the challenge is that what is the subset of single cells we need to use? And that subset could be different between different pairs of genes too. So right now we have a method that is able to handle this kind of situation. So stay tuned. So finally, I do want to acknowledge all of the key contributors, to this work. So this is a joint work with, like I said, so this is our biology collaborator, and also my senior colleague, Peter Bickel. So we worked very closely on this project, and this is a postdoc, working with 3 of us. Thank you.
I think we have time for a couple of questions. And could you repeat the question so that it gets recorded
Sure.
properly? So.
June?
So that when you collaborate with biologists, typically they would like to know how reliable of your. So then I wonder, how do you? Typically, people are not just satisfied when you're giving them a list, but they would like to know how reliable, and before they decide whether they go for experiments or not. So how do you address that?
Yeah. So the question is that, when we are collaborating with a biologist, and when we have delivered some prediction results to the biologist, how we can convince the biologist to do some follow-up experiments, right? So I think this is relying on a lot of different factors. So one thing is that, who is your collaborator? So I found that, building some trust, with your collaborator, that's very important. And for this collaborator, we have worked together for about, 10 years. So we know each other well, and we trust each other well. And also for this analysis, so like Marissa Medina, she has been deeply involved, so through the whole process. So this project actually took longer than I have expected, almost 3 years. And through that process, Marissa was very patient. And we look at all of the results of figures together. So she knows the process, the trouble, all of those. And so therefore, she was convinced, actually more than us, when she was, seeing the results. And she, immediately said, I want to do experiments on that genes. Yeah.
As you can see, the classes are changing, but maybe you could stay for a few minutes. If there are other questions, you could come forward and have a little discussion. Thank you again for a
My
great
pleasure.
talk.
Thank you.